Hi, and thank you for joining me today while we extract DNA from a strawberry. The way this works is you take soap and you break down the cell membranes and cell walls and you pour alcohol in the solution to pull out the DNA so the solution falls out. So your first question might be, what is DNA? DNA is the hereditary material in humans and almost all other living organisms that makes us different. It gives us characteristics that are unique to each individual, and it's what makes you and I different from each other. So the first thing we're going to do is break down the strawberry. So you grab a nice big strawberry and you put it in a bag, and make sure the bag can zip closed because you're going to put water and then you're going to smush it all up. And make sure to try and smash it as much as you can because you don't want any hard parts left. You want it to be very soupy. And what we're doing now is breaking down the cell membranes and the cell walls so we can get to the nucleus. And inside the nucleus is where the DNA is, and that's what we want to get to. So for the most part, we have a pretty soupy mixture. And now what we're going to do is take it and pour it into this clear cup. It's important to make sure your cup is clear so you can actually see the DNA when you're done. A coffee filter or some cheesecloth can be used to filter out the hard parts of the strawberry that we no longer need. Okay, so now we have our strawberry soup mix, and in here we have broken down all of the cell membranes and the cell walls, and so now we have to break down the cell or the wall of the nucleus. And how we're going to do that is take some dishwashing detergent, and this reaction works because the dishwashing detergent breaks down the lipid layer of the nucleus. So when you wash dishes, you're washing grease and stuff off of pots and pans. So it works the same way. So you take some and just pour it in. And then you take a spoon or a measuring utensil to stir it up. And you want to make sure to get it really foamy. So now it's nice and mixed up, and all of the lipid layers of the nucleus have been worn away. So now we can actually extract the DNA. To extract the DNA, we're going to use isopropyl alcohol. This works because it pulls the DNA out of the solution because DNA doesn't dissolve in alcohol and all the other parts of the solution do. So it's going to separate, kind of like how oil and water separate. You want to pour it in very slowly. Alright, and as you can see here, there's the top layer with the alcohol and the bottom layer with the soupiness. And in the top layer, all those little spindly white cloudy things, that's your DNA. And if you wanted to, you could pipette it out and put it into a vial to keep. But that's it. There's your DNA.